We're getting started today, so you have your homework due. Please don't forget to put that in the folders before the end of class. Start today with a couple of announcements. The first one, so this is a fairly serious announcement, so please everyone pay attention. We've had a couple of incidences on campus recently, some very near the biology building. I want to make sure, particularly for those of you that have late labs, that you know your options for staying safe. All right? You're going to see this in lab as well because we want to make sure our students are safe. So you should be aware, so just particularly if you're on campus late at night, be aware of your surroundings. If you see something happening, call the police. But you do have a couple of options for if you have to walk across campus late at night. There's an escort service on campus that you can call that will walk with you. You're alone. If you're leaving, if you're in one of the later labs and you're leaving after dark, maybe wait for a friend and go together. The labs are going to talk to you about some safety precautions we're going to take in the labs later in the afternoon and in the evening. There's also a Guardian app, which I think you can check in and say where you're leaving and where you're going. And if you don't check back in, they'll come looking for you. So you have some options to stay safe on campus. All right, so just be aware. There have been some incidents recently. Everyone, be aware of your surroundings. Take precautions if you don't feel safe. If it's late at night, you have some options. Okay? Everybody stay safe. All right. The next announcement about the repolling, I've been getting some emails, and I just want to clarify one more time. For the repolling, you get a trial period of a couple weeks. Once that is expired, you have to pay them to continue to be able to see the snapshots of the slides after class. You do not have to pay them to continue to use your iClicker. All right? The only people who need to pay for the repolling registration are those people who are using mobile devices. You do need to pay them. But if you are using an iClicker, as long as you are registered on the repolling, I will still get your responses. You just can't access that snapshot view with the correct response after class. Which means you just need to take notes on what the correct response is because I put the slides up on LaLima and Facebook. Right? Was that clear? The only people who should need to pay for the repolling are those who have decided to use a mobile device to respond. Everybody else, you don't need to pay unless you want to. I'll still get your responses. Okay? Any questions about repolling? I'll be posting everything today. So you guys should thank your TIs in recitation this week. They came and helped me grade for almost 4.5 hours last night. All right, so they were there grading with me until after 9 o'clock. But we got them all done. All right? And the TIs have your exams, and they are entering your grades. And you'll get them back in recitation this week, so you get a chance to look at them and ask questions. I'm waiting for 1 or 2 more TIs to get their grades updated on our grading sheets, and then I'm going to post them all on LaLima by the end of the day today, I'm hoping. Hopefully earlier, but certainly by the end of the day. Along with that, I'm going to post your current up-to-date homework grades, quiz grades, and participation points so that you can check carefully and make sure that you're getting the participation points that you think you should, or that they're being logged to your name, that we haven't made an error somewhere in entering your quiz or homework grades. All right, so go through that very carefully, make sure everything looks correct. As far as the exams, you'll be getting them back Thursday and Friday in recitation. We have a chance to look at them and ask your TIs some questions to explain what questions you missed that you don't understand the answers to. If you are looking at your exam, and again, remember there are 8 of us that are working together to grade your exams. Check the math carefully, make sure we didn't make a mistake. If you find a mistake, bring it to me, okay, and we can correct that. If you have a question about the way something was graded, all right, so I'm going to give you, today is Wednesday, I'll give you 2 weeks from now to come in and talk to me about, you can always come in and talk to me about comprehension, but if you're going to come to me and ask me about getting points back on something, you've got a 2-week window from today, all right? the grades are set. So we can start thinking about the next exam. All right, and I'm doing that just to make sure that someone doesn't come in at the end of the semester and say, I found all these mistakes on my exams. Right? I want you to look at them now and understand where you made your mistakes. Right? And if you feel something was graded incorrectly, you have a 2-week window to bring it. You can always come in and ask me questions about you don't understand what the answer is later. But for the points, 2 weeks. Okay? Is that clear for everyone? All right. So look for those. As soon as I get them posted, I will send out an announcement so that you can all go and check them immediately. I know you're anxious to look and see how you did. We'll get them to you as soon as I have access to all of them. Okay? Any questions about the exam or the grades or anything else related to that? There won't be a key posted, but you'll have a chance to discuss everything in recitation, both this week and next week as well. So you'll get some time in recitation this week to go over, look at your questions carefully, and then have some time to ask your TI to go over those questions and why, what the correct answer is, and why. So you have plenty of time in recitation to do that. Any other questions? All right, I think that was the last announcement. Oh, so been having clicker issues, connectivity issues. We'll see today if the machine is going to work or not. We'll start off with the clicker questions to find out. All right, so this is a review from last class. We're talking about what a gene is, the components of a gene.
The arrow has to be pointed the opposite way.
We're talking about a promoter, the directionality of a promoter, what it does, and how it determines what strand serves as a template. Just to get everyone's brain turned on, which strand is the template strand in that diagram? Alright, everyone get a chance to get their response in?
Oh, how did I say that? Oh, my Pilates? Yeah, it didn't work for a while.
Alright, let's take a look and see what you think.
I did that.
Alright, so it looks like class, majority says the bottom strand. Alright, so if this is the promoter, which way is transcription going to happen? To the right or to the left? To the right. So it's going to transcribe like this. So which strand is oriented opposite to that?
Bottom one.
Bottom one. All right, that's correct. All right, good. Everyone remembers what we did last time? And back to, there we go. All right, so we're going to continue on. We're talking about the central dogma. We're in that process of transcription where DNA makes RNA. All right, so we haven't honed in on mRNA yet. We're talking about what a gene was, and we were defining the components of that gene, the anatomy of the gene, if you will. We talked about the limit, the transcribed region, and the promoter on Monday. So if we looked at that transcribed region, the transcriptional unit, notice it is not as big as the gene, so it doesn't extend all the way out to the gene limits. How does the polymerase know what that transcriptional unit is? How is it defined? This isn't, not trying to trick you. All right, what has to be in the sequence? An initiator.
Promoter.
And a.
Promoters.
Oh, we're talking about the promoter. We're just talking about the part that's just transcribed. How does it know where to end?
Terminator.
Right, a terminator. So there's got to be some sort of sequence in there that says start here, end here. Do we have an initiation point? And a termination point. Right, and that has to be determined by that sequence. All right, so we're almost there as far as the anatomy of the gene. We have just a few more things on here that we haven't talked about. What about the bits that are between our promoter and transcriptional unit and the ends of the gene? Why are we including those as part of our gene? Right, we've already got a transcriptional unit, start and stop site, making a product. Why do we care about the rest of that sequence? Is it part of the gene? The answer is yes, since it's on the slide. All right, what does it do? Any ideas? Regulates gene expression. Thank you. So we've got regions that are going to determine the expression patterns of our transcriptional unit. All right, so there's our anatomy of a gene. We have 6 components. We have the upstream regulatory region, the promoter, the transcription initiation site, the transcription unit, the piece of DNA that's actually copied into RNA, transcription termination, and then the downstream regulatory region. This is what makes up our genes. So when I say draw a gene, I want to see these 6 things. All right? All right, if we zoom back out and look at multiple genes on a chromosome, let's think about the way transcription happens. So as it's drawn, are all of these genes transcribed in the same direction?
Vikings did that.
I know, but what would you want to do that?
I don't know.
You think the last person ever expected it?
All right? This one should be pretty straightforward.
My mom has that.
Take a look. Right. Right. Based on the transcription, the blue wiggly worm, and the direction it's going. Right, so they're all going the same direction. What about now? Are they all transcribed in the same direction as I've drawn it?
No. He does that.
All right, again, pretty straightforward. Take a look.
Banana.
All right, correct. As I've drawn it, they're going different directions.
Como.
What about 7 genes per chromosome. So I haven't drawn anything. What happens in reality? Are all the genes on a chromosome transcribed in the same direction or not?
I'm gonna Google this. No, because it's a gene is a certain region, right? You don't, like, it could go in the opposite region. You know, like, if it's in the opposite gene, in a different gene, it could go in the opposite region, right? Yeah. I want to test Google.
All right, everyone get a chance to answer? Let's take a look.
Yeah, I can draw. That's what I'm saying. No.
All right, so a little bit more split decision. Some say yes, some say no. All right, the genome is extremely complex. Some genes have their template strand on one strand. Some genes have the template on the other strand. Genes can have overlapping transcriptional units. The transcriptional unit of one gene can be in the intron of another gene. All right, it's a complex system. We're drawing it as a very simplified system so we can understand how all the parts work together. But I want you to, we'll talk about more of it as we talk more about transcription and get into some of the processing of mRNA. But I want you to recognize that what we've drawn is a simplified version of what's going on in the genome. Genes aren't necessarily one nice neat unit surrounded by a promoter, then there's some space in another gene. They're all mixed up together, right? Templates on different strands, overlapping, etc. All possible ways you can think about it being put together. All right, so that's what our gene looks like in the simplest sense. We have some regulatory regions. We have a promoter that's going to bring in and bind the RNA polymerase. Then we have a stretch of DNA that's going to be our transcriptional unit that serves as the template for that polymerase to make an RNA copy of our DNA template. So we know that transcription involves an RNA polymerase already. We know that polymerase, like other polymerases we've talked about, synthesizes, in this case, single-stranded RNA in a 5' to 3' direction. And that we're making RNA from a DNA template. All right, so let's take a look at the 4 steps of transcription. All right, when we talk about transcription, we talk about translation and some of these other processes in the cell. The steps are going to seem pretty familiar. We have to bind the relevant enzymes. We have to initiate the process. If we're making a product, we have some phase of elongation where you're actively making that product, in this case an RNA. And then you have to have some way to end the process. Pretty straightforward. You could use that to describe almost any process where you were creating something. We look at it in the context of transcription. We have our chromosome. On our chromosome is a gene. There's a promoter on there somewhere. We have to bind our polymerase. All right, for the binding step, and we're going to look at next more closely how this happens. But just generally, first we bind it to our promoter. There's an initiation step. All right, one of those strands is a template. Think about the process of replication. We need a single-stranded template. So we have to initiate that process and create a transcription bubble. So we have to pull apart the double-stranded helix so that the polymerase can get to the template to make a copy. Oh yeah, just in case you weren't sure yet. Try it one more time. Which strand is the template strand? All right, everyone get a chance to answer that one? We've done lots of practice on this one, so no one should miss this one on the exam, right? Let's take a look. All right, correct. It is the bottom strand. We are transcribing to the right, 5' to 3'. The oppositely oriented strand is the bottom one. All right, so we bind, we initiate. There has to be some signal that says everything is ready, go. Start transcribing. Then we have an elongation phase, and this is simply the phase where we are actively making that RNA transcript. So you can see there that triphosphate on the end of our nucleic acid. That transcription bubble, right, moves down the chromosome with the polymerase, ahead of the polymerase, as the polymerase copies the template strand, and it reforms behind it. So we get our RNA transcripts being generated, 5' to 3'. And then at some point, there is a signal in the sequence that says, this is the end, stop transcribing, polymerase falls off, the transcript falls off, the double helix reforms, and we have our original chromosome, and now just a transcript of the gene that we were expressing. All right, so those are the 4 stages: binding, initiation, elongation, and termination. If we think about the expression of genes in any one cell, is the gene always turned on? All right, so what does that mean about the expression of genes? It varies in what? Time. Right? Gene expression for a particular gene varies in time developmentally. In any one cell type, is a gene either always on or always off? Can you think of a gene that might be expressed in a particular cell type? What defines a skin cell from a muscle cell? No examples? Can you think of genes that are expressed in all cells? This one you should be able to think of. All right, what sorts of cellular products are needed to keep a cell alive? What makes proteins? Amino acids. What's the organelle that makes proteins? What makes proteins? You guys, thank you. The ribosomes. What are ribosomes composed of? Ribosomal RNA. Where does ribosomal RNA come from? It is from a gene. It's transcribed. So all cells have to make ribosomal RNA. Otherwise, they can't make protein. All right, so there are differences in when and where cells are expressed. We've looked at the process of how they're transcribed, that first step in the expression process of turning a gene into a product. And more specifically, we're going to start talking about now turning a gene into mRNA and mRNA into protein. So just to give you some examples, there are differences in expression in space. So if you look at a Drosophila melanogaster, so the fruit fly embryo, Each one of these panels is the gene expression of a different gene in an embryo. Where it is purple is where the gene is expressed. So just to give you an idea of the variations in gene expression patterns, right, at one point in time during the development of a fruit fly. So lots of genes are expressed in very different patterns. Here's an example from a vertebrate. We're looking at a mouse embryo at 11 and a half days. We have myogen being expressed just in the muscle tissues. Right, variations in space as far as gene expression. And we see developmentally a lot of expression gradients. So here's a schematic again of that Drosophila embryo, where on the anterior end you get the expression of one gene, and the posterior end you get the expression of another, and there's a gradient in between. Right, that helps set up these developmental fields. So there's also variations in expression in time, like we talked about. So if you look at the embryo from early to late development of that embryo, different genes are turned on, depending on what stage you're in. And the expression patterns of these genes, particularly in development, right, we see these developmental gradients through space and time, and they help determine what segment becomes what part of the fly. All right, so expression patterns are important, and when and where they are turned on and off do important things. All right, so the next thing we're going to look at relative to transcription, we're going to get into the control of transcription. What's controlling when and where these genes are expressed? All right, we're going to look at a little more detail, that process of transcription and how it starts and what's controlling that. So we're looking at the regulation of transcript levels. So if we wanted to control the expression of something, how would we do it? What stages of this process might we try to target?
Binding process.
The binding process of what?
The RNA polymerase with the motor.
Yes. So we could control transcriptional activity by controlling binding of the polymerase. What else could we do to control expression levels? What other part of the process? What happens after we've made a transcript? We've got some RNA floating around. Sorry? No?
Control the amount of time the RNA strand is produced.
You can control sort of the volume of transcripts produced. So how long are you transcribing that gene? Any other ideas? You can also control it, so if you're transcribing a gene, RNA is inherently not all that stable. So one way you can control it is so these are all sort of pre-transcriptional or during transcription processes. You can also control it post-transcriptionally by just degrading that RNA as it's being made. All right, that has to do with RNA stability. But most of the way that we're controlling gene expression are exactly what you've come up with. These ways to control transcription. All right, so we're going to look at the process of transcriptional control. First, in prokaryotes, we're looking at the classic lactose operon, which some of you have hopefully heard of or read about in your book. And then after that, we're going to move into looking at the eukaryotic system, which is a bit more complex. We're going to start off with prokaryotes. We're looking at an operon. So what's an operon? Define an operon. Anyone know? Has to do with transcription. We're talking about the control of transcription. I'll give you a hint. No guesses? Right, an operon is a suite of genes that are under the control of a single promoter. Generally related in function. So think about prokaryotes, single-celled. They don't have a lot of time to respond to changing environmental conditions. If you have your genes grouped functionally, and you move into a new environment, you can turn on all of the genes at once to deal with whatever that new environment is. So let's take a look at the lactose operon. Lactose operon confers to that prokaryotic cell the ability to grow on lactose. Seems like an advantage. Gives it an alternate source of food. What is lactose? Oh, before we get to what is lactose, let's look at the lactose operon. All right, so here's our gene that we pieced together over the last couple of lectures. Here's the transcriptional unit, our promoter. Within this transcriptional unit, we have 3 different genes. The LAC-Z gene, which we're going to look at in a moment, which is a beta-galactosidase. The LAC-Y gene, which is a permease, which is a membrane protein that, when turned on, the protein moves to the membrane and lets lactose through. Right? So it serves as a pore for lactose to pour into the cell. An opening for lactose to come into the cell. All right, and those are the two, LAC-A, I think, is an acetyltransferase, also related to lactose. Let's take a closer look at LAC-Z. So this is lactose. All right, so we have galactose and glucose linked. The beta linkage. Right, because the hydroxyls are in opposite orientations. Makes it a beta linkage rather than an alpha linkage. And that LAC-Z gene in particular, right, so it makes this molecule a beta-galactoside. And the LAC-Z gene is a beta-galactosidase. All right, so LAC-Z, when present, is going to cleave that beta linkage and turn lactose into galactose and glucose. What is glucose to a cell?
Sugar. Energy.
Sugar. It's energy, right? So by turning on this LAC operon in the presence of lactose, the cell can quickly get access to glucose. All right, so the lactose operon is inducible, which means that it is only transcribing, excuse me, only expressed, only making those enzymes in the presence of lactose. So when there's lactose around, it turns on the operon, it makes LAC-Z, the beta-galactosidase, which can then cleave that beta linkage, which produces glucose for the cell. Seems like a pretty good idea. All right, let's take a closer look at how that actually happens. So we have 2 factors at play here. We have the lactose, which is the inducer of the operon. So when lactose is around, the operon gets turned on and those genes get expressed. And then we have, so the levels of lactose are important, and then we have glucose. The levels of glucose are important. And we'll see that the presence or absence of these 2 things, so glucose helps turn off the system. So when you don't have any glucose and lactose shows up, you turn on your operon, you can make glucose. When you've got enough glucose, then the system will be turned back off. All right, so let's take a look at that inducer first. All right, if we look here at our promoter, our operator, and then our 3 genes in that operon, we have a regulatory gene somewhere upstream that's always expressed. That's our repressor. So when this is present, it binds to that operator, and the operon is not expressed. In the presence of lactose, lactose binds to that repressor and inhibits it, so it can't bind to the operator. The operator is now free. The RNA polymerase can bind to the promoter, and the operon is expressed. All right, so it's through the direct interaction of that lactose binding that repressor that operon gets turned on. So that's how we turn the system on. All right, just to remind you, that again is what that lactase is going to do. It's making glucose. So once it's turned on, it starts cleaving lactose, starts producing molecules of glucose, and then as the glucose concentration in the cell increases, that helps shift the system back off. And it does that by interacting with this cap, which is a cyclic AMP activator protein. Right? So in order for the RNA polymerase to bind and express that operon, cap has to bind to the promoter as well. So in order for the operon to be expressed, cap has to bind, and the repressor has to not be bound. In the presence of glucose, that interacts with cap, and cap can't bind, and transcription doesn't happen. So it gets shut off. So as glucose levels increase, the system gets turned back off again. All right, so we have the levels of lactose and glucose interacting to form a shifting system to turn on that operon only when needed. So the cell isn't wasting resources making genes it doesn't need. All right, so let's look a little more closely, right? The lac operon be a little bit confusing. So let's look at all the possible combinations of glucose and lactose and whether that operon is active. All right, so we have over here, I'll indicate whether glucose and lactose are present or absent. We have our cap binding site, our promoter, our operator, and our genes. And we have our cap activator and our lac repressor. All right, all the components of this system that help determine whether the genes are being transcribed or not. All right, so let's look at all possible combinations of these things. So first thing, we have glucose present, we have lactose present. Is the operon being transcribed? Talk it through with your neighbors.
Glucose is present, right? So you don't need the lactose.
That's for yourself.
It only turns on when there's no glucose. Okay, because there is glucose, you don't need that.
No, it's not. Okay, well, something about when there's high levels of glucose, when there's high levels of glucose, then I don't know. I forget the rest. I just heard that. Right, because lactose is
About what glucose does and what lactose does. Let's take a look and see what you guys think. All right, so somewhat of a split decision. Let's walk through it. All right, so in the presence of lactose, what happens to the lac repressor?
It gets bound by lactose.
It's bound up by lactose, which means what?
It
allows
removes
to
from
use
the
it.
binded site.
It can't bind to the DNA, which means that
The operand is on.
the promoter is free
To bind.
to bind to the polymerase. What else does the polymerase need in order to bind to the promoter?
The cap activator.
The cap protein, cap activator. What happens to the cap activator in the presence of glucose?
It comes off.
It comes off. Alright, so in the presence of glucose, so in the presence of glucose, although the lag repressor is bound, so is the cap activator.
What?
Give me button. There we go. So it is off.
Wait, I'm so confused.
So it's off.
Alright?
No, it should be.
Glucose is present at high enough concentrations that cap activator can't bind, which is needed for the polymerase to bind. Alright, we'll do one more and then we'll finish on Friday. Okay? Alright, so second situation. We have glucose present but no lactose.
We don't have lactose.
Is the operon on or off?
Oh, no, because there's no lactose, right? If there's no lactose, your lactose repressor is still on. The repressor.
After the lag.
No, you still have the lag. If it's lactose, if there's glucose, it's off. Yeah, if there's glucose, it's off. But if the lactose isn't there, the repressor is still on, and it's off. There's no lactose.
No, because there's received I don't know. Like,
Okay, hey.
so like what makes like yeah, so it's like inserted
Middle one? Rob Oda? Right there, yeah.
and that's when you
Thank you.
Alright, so there's no lactose. The repressor is bound. With glucose present, both of those things are working in concert to make sure that that operon
yes,
is not
I still have to
going to be expressed. Alright, so we're going to stop there. We'll pick up here on Friday.